Well, guys, in these slides, we are going to discuss the genetics development of Arabidopsis Thaliana flower. And you know that this is the one of the model organisms which has been considered in the biological labs for the different kinds of the studies. It's a simple model organism where we can study the different aspects of the biology, and also we can study different aspects of the genetics. So here we further proceed to study the development of Arabidopsis thaliana flower. Before to the specific study on the flower of Arabidopsis thaliana, we will discuss the normal appearance and also the development of the flower in common to all plants. Cells in developing organisms do not only differentiate, they differentiate in different patterns. Now here, if you are going to consider the developing organisms are the multicellular organisms. In multicellular organisms, the cells not only differentiates, it differentiates into the different patterns. Now if you are going to consider any plants, you consider a kind of the species with the other, there will be the difference in the appearance of the cells, not only in the differentiation of the appearance, it also seen that there will be the defined patterns of that particular differentiation. So here, if you are going to consider example the rose plant, and also if you are going to consider the lily plant, these two different plants which are going to have the differentiation, but when it is going to differentiate completely, the patterns are going to differently appear. So in the sense, the leaves of the rose plant and the leaves of the lily plant is going to appear different, and also the whatever the flowers are going to present, they are going to appear to be different. In the same manner, within the plant, the organs are going to be the very different. For example, if you are going to take the flower within the plant, like a rose plant, the flower, the differentiation is very different from the what the leaf is, and also the patterns also. Now here, the appearance of the flower on the stem is also going to differentiate it. So here, this is what the meaning of this line. So further is a best example is the differentiation of flower. In most plant families, flower consists of four types of organs: sepals, petals, stamens, and carpels, each composed of characteristics cell types. Now here, flower is composed of four different organs, that is the sepals, petals, stamens, and the carpels. They are mentioning these to be as the organs because they have their specific functionality within the complete flower. Now here, if you are going to consider the sepal is going to be different from the petal, and the same manner the petal is going to be different from the stamen will be different from the carpel, and all these four having the four different patterns compared to each other, and also the four different functions compared to each other, and also the four different cell differentiations. The patterns which appear in the flower is the outermost hole of the flower with the sepals, the petals next closest to the center, the stamens even closer to the center, the carpels in the central region. So these are the four different organs which has been arranged within the flower. In developing plants, determine their position, and in response to this determination, differentiate to the cell types appropriate for that position. Now here, this line is very comprehensive. In this the meaning is that the developing cells are going to position the flowers, and in response to this determination differentiate to cell types. So here in response to this, the whatever the pedicel is going to develop, and through that pedicel whatever the sepals, petals, and the stamens and the carpels are going to develop, that will be determined during the differentiation, and the appropriate cell types are going to be positioned during this differentiation. That is the uh, meaning of this particular sentence. The every species of flowering plant, the disposition and number of these organs is also specified, and the floral formula is repeated in each of the flowers on each individual plant of the species. As I have already spoken, the difference between the rose and the lily. So that is like in every species of flowering plant, the disposition and number of these organs is also specified. Like for example, if you are going to take a rose, in rose the sepals are few, petals are many, it is polypetalous, and also the stamens are many, and the I hope the rose is a bicarpellary in its ovary. Now here, when you are going to consider the lily is different from the rose. Rose is having the different flower structure compared to the lily, where lily is going to have pentapetalous and hopefully the zygomorphic kind of thing, and pentapetalous, pentasepalous, and also it is going to have the 4 stamen, I hope to 4 or 6 stamen, and the carpel. So the difference between the lily flower and the rose flower has been differentiated, and this difference within this species, I mean the common appearance of the structure of the flower within this species is going to be hold good from one generation to the other generation, and there will not be any change in the pattern of its development, let it be in the rose or in the lily or any species, let it be any species. That is what the meaning of this sentence. So there will be 3 classes of genes controls the basic patterning of flower organ development. These 4 organs development will be controlled by the 3 different group of genes. Those are organ identity genes, which helps to induct the development of the different floral organs. That is the different floral organs, or that is the 4 organs. That is the petals, sepals, stamens, and carpels. Cadastral genes will be responsible for specifically to define the boundaries, that is the expression of the organ identity genes. That is, cadastral genes are going to control the organ identity genes to make the specification between the cells differentiating for the sepals, and the cells differentiating for the petals, and the cells differentiating for the stamens, and the cells differentiating for the carpels, so that these 4 different cells should not get mixed up. So the patterns are going to be remain very specific, creating a defined boundary. Next is meristem identity genes. These are going to differentiate vegetative shoot meristem to inflorescence meristem cells. So here vegetative shoot meristem. So this vegetative shoot meristem, in the sense they will be totipotent in their nature. They can differentiate to any part of the plant. So when meristem identity genes are going to express, these genes are going to express at a particular location within the plant. At that position, if there is a need of the development of the flower, at that position they are going to develop into a inflorescence meristem. In the same manner, these vegetative cells will be responsible to get differentiated into the different organs of the plant. But as per our discussion now in these slides, it is very much clear that the vegetative cells are going to, or the vegetative shoot meristem is going to differentiate into the inflorescence meristem cells. So next is Arabidopsis thaliana has been extensively studied as a model for flower development. The developing flower has four basic organs: sepals, petals, stamens, and carpels. As the symmetry of the flower is actinomorphic, the organs are arranged in a series of holes, where four sepals on outer whorl you will find in the flower, four sepals on the outer whorl, four petals in the next whorl, six stamens in the third whorl. A central carpal region is fourth whorl. So it means the outer whorl is the first, second, next whorl is the second whorl, and the six stamens in the third whorl, that is the third whorl with stamens. Carpels will the fourth whorl. So that is the structure of the flower. Organ specification in flowers with certain genetic evidence. Here, a preliminary model based on experiments with Arabidopsis thaliana is observed. The model is by and large consistent with existing evidence and has predicted the result of a number of genetic and molecular experiments that have been recently performed. So here, on the basis of the experiments conducted with the Arabidopsis thaliana in regarding with the flower development, there is a massive observation of the flower development. So according to that, the genetic note has been taken down. So there is the ABC model of flower development by E. Soin and the E. Merowitz. These are the two persons who have developed this ABC model of the flower development. According to this model, floral organ identity genes are divided into three classes. According to this ABC model. Class A genes, which affect sepals and petals. Class B genes, which affect petals and stamens. Class C genes, which affect stamens and carpels. So here, these three classes having the combined expression, where this expression is overlapping on the two different adjacent organs developing process of the flower. Now here, if you are going to observe the image, in this you can see the class A gene and the class B gene and class C gene. These three genes have different shades. Class A gene is responsible to development of the outermost whorl, that is the first whorl of the flower, that is the sepal. Class B gene will be responsible for the development of the second whorl and the third whorl. Third whorl in the sense stamens, second whorl in the sense the sepals. But the class A gene is going to influence on the development of the class B gene, third whorl, that is the sepals, and class C genes will be responsible for the development of the fourth whorl, whereas it is going to get influenced by the class B gene third whorl development. Now here, this is how there will be the influence of the class A, class B and class C genes organ identity. So here, as we have discussed, class A genes affect the development of the sepals and the petals, and class B genes which affect the petals and the stamens. Now here you can see in the class A and class B, the common is the petals. So the influence of the class A and class B genes are going to be on both influences of class A and class B genes are going to be on the petals, that is the second hole from the outermost. Now when you are going to see the class B and class C genes, the stamens are the common. So class B and class C genes are going to have the influence on the stamens. So this is what the combinational effect we can say of the organ identity genes as per the classes A, B and C. These genes code for transcription factors that combine to cause tissue specification in their respective regions during development. Transcription factors are very important during the development. Those transcription factors will define the cell specificity of the organ. Although developed through study of Arabidopsis Thaliana flowers, this model is generally applicable to other flowering plants. So here, with the help of the Arabidopsis Thaliana flower, this particular classes has been studied. So this model is generally applicable to other flowering plants also. So that is what the meaning here. So coming to the next slide, flowers develop groups of undifferentiated cells that form on the sides of the inflorescence meristem. These cells must differentiate appropriately into various floral organs. So here the meaning is that whatever the flowers are developing from the undifferentiated cells is nothing but the other shoot cells, which we can say that multipotent or totipotent. So here many shoot cells will be totipotent under the influence of certain external factors or the induction of the adjacent tissue type or the organ type is going to develop into a particular required organ at that particular position. So here, as we are discussing about the flower development, so here the undifferentiated cells are which are present at that particular location within the development of the flower inflorescence meristem, which is going to differentiate appropriate into the various floral organs. So here further we are going to study the Arabidopsis thaliana mutants. So here from here we are going to study how the differences are going to appear within the development of the Arabidopsis thaliana flower. The floral organs are formed in holes which they do not occur in wild type flowers. So here when you are going to consider the mutant, as we have already discussed that the flower is having the 4 different organs. Now here, as per the first point, the floral organs are formed in holes where they do not occur in wild type flowers. Wild type flowers, we discussed that the 4 organs develop, that is the sepals in the outermost, petals in the next, and in the third hole there will be the stamens, and in the fourth hole there will be the corpus. So this appropriate positioning is not going to happen. That is what the meaning within the mutants. The mutated organ identity gene will always affect the organs of 2 adjacent flowers. So here, if you are going to consider the organ identity genes, which has been already classified here as ABC, and it is shown here in the diagram. And remember guys, this is the A group, B group and C group, and we have discussed that class A group genes are going to have influence on the B group genes in the same manner, vice versa, and B group genes are going to have the influence on the C group in the same manner, the vice versa. That is, the C is going to have on B and B is going to have the influence on the A. So the A class A genes is going to be responsible for the sepals development and the petals development, but with the combination influence of the A and B for the petals. And whereas the class B genes is going to have the combination development of the stamens with the class C genes. So these two are going to be influencing, whereas the carpels will be specifically taken care by the class C genes, and whereas similar to the class A genes, which will be responsible for the development of the sepals. So in this manner, this combination is going to express its identity. So if you are going to mutate any of this, A class A genes, B genes or C genes, will always affect the organs of two adjacent wholes. So here if you are going to mutate this particular class A genes, so class A genes will not appear to develop their organ, whereas there will be the influence of the class C genes and the class B genes. So there can be the development of the class B and class C organs in the whole 1. And in the same manner, the recessive mutated organ identity genes will always be suppressed by adjacent whole developing organ identity genes. As I have said, that if you are going to mutate class A gene, the mutated class A gene is going to act as a recessive gene. Because of its recessive expression, the next wild type, whatever the unmutated or the wild type or normal gene is present in the B and C class genes, is going to be the dominant over the mutated class A gene, that is the class A organ identity gene. So because of that, the two adjacent wholes are disrupted within the flower primordium. Here flower primordium in the sense the inflorescence meristem, which will be responsible to develop to the 4 different organs of the flower. Hence another extra hole or 2 similar other holes will develop within the flower. So here if you are going to mutate one of the organ identity genes, that mutated genes will not develop into their organs, but the other organs can influence at that part. For example, if you are going to mutate class A genes, the carpels can develop at this sepals position. Because of that, there will be the 2 holes of the carpel. If you are going to mutate the class B genes will be responsible for the petals and the stamens. At this position, there can be the development of the sepals of one set extra and the carpels of the one set extra. In this manner, the extra hole of the similar organ is going to develop of normal genes of the organ identity class. So this is what the meaning. So here further, these organ identity genes will be controlled by the homeotic genes. So here homeotic genes controls the organ identity in Arabidopsis Thaliana flower development. So here these homeotic mutants has been discovered in the development of abnormal flower. Abnormal flower in the sense mutated flower, where during the experiments they are going to mutate the genes to study what exactly the mutated genes will be responsible. For example, as we have discussed that in the class A genes there will be the development of the petals and the sepals, and class B genes it will be responsible for the development of the petals and the stamens, and in the class C genes it will be responsible for the development of the stamens and the carpels. So here they are going to mutate those genes, this group of genes, and they are going to study. According to that, they are going to make a note and declare it to be as the so and so genes, which is going to be responsible within that particular class. Now here homeotic genes are the one which are going to control the organ identity in the Arabidopsis Thaliana for flower development. Now here the roles of 4 genes of this homeotic genes. Those genes are the APETALA2, APETALA3, PISTILLATA and AGAMOUS. These 4 are corresponding with the sepals, petals, stamens and carpels. These are the 4, in which recessive mutations remove the normal roles of each of the genes in floral organ development. So here, if the normal gene has been mutated, that particular mutated gene is not going to express in the form of the normal development of that particular organ. That is what the meaning. Mutations fall into 3 classes of function genes. Now again, these mutations has been classified corresponding to these groups, but it has been mentioned as the function genes. So here what the classes has been mentioned here is class A genes for the organ identity genes, class B genes for the organ identity genes, and class C genes is for the organ identity genes. Now here, as we are discussing the homeotic genes controlling the organ identity within the particular flower development, they are being corresponded with the ABC model of the flower development of the organ identity genes with the homeotic, where they are going to call these classes as, so the classes are function A genes, function B genes, function C genes. In the function A genes, there will be the APETALA2. In function B genes, there will be the APETALA3 and PISTILLATA, and function C genes, there will be the AGAMOUS. So these are the 4 homeotic genes which is responsible to control the organ identity during the development of the Arabidopsis thaliana, is grouped into function A, B and C classes. Now here you can see there is this is the wild type flower. You can see this is the mutant APETALA2 and this is the mutant APETALA3 and PISTILLATA. This is the mutant AGAMOUS. Now here, when you are going to consider the wild type, you will see sepal, petal, stamen and carpel. So this describes the organ identity. So here in the mutant APETALA2, you will see the carpel, stamen, carpel. So here you can see the flower structure has been got abnormal because of the mutation in the APETALA2. So here you can see the mutation in the outermost 2 holes, outermost hole and the outermost next hole, that is of the sepal and the petal. Now there is the APETALA3 and PISTILLATA. The petal has been disappeared and the stamens has been disappeared. That is the APETALA3 and the PISTILLATA. Now in the APETALA2, there is the disappearance of the both sepals and petals. But in the APETALA3 and PISTILLATA, there is the different disappearance of the petals and the stamens. In the AGAMOUS, there is the disappearance of the stamens and the carpel. Now this is what we have to understand in the next slide. What is the mutation exactly? Now mutation in the type A genes, that is the function type A genes, is that affects the sepals and petals, which are the outermost holes, that is the outermost first hole and the second hole. In this mutant, the gene is APETALA2. Carpels develop instead of sepals and stamens in place of petals. Now here, this is the APETALA2 mutant. When you are going to mutate the gene APETALA2, there will be the absence of the sepals and petals. So here APETALA2 falls into the function A genes. So here class A genes of the organ identity. Now here this organ identity class A genes will be responsible to development of the both the petals and the sepals, the outermost sepals and the petals. If you are going to mutate, the differentiation is not going to happen. Because of this, the absence of the induction of the differentiation by this particular class A genes, when it is mutated, the apetala2 will not express its normal function. As it is going to be recessive, it is going to give the chance for the development of the other two oles, that is the third and the fourth stamens and the carpels, in the place of the sepals and the petals. So here you can see carpel, stamen, stamen, carpel, carpel, stamen, stamen, and the carpel. That is what the formula changes from the wild type. So here carpel, stamen, stamen, carpel, carpel, stamen, stamen, and the carpel. In the wild type it is not so. In the wild type you can see the sepal, petal, stamen, and the carpel. But in the apetala2, you will observe carpel, stamen, stamens, and the carpel. And the other part is going to be in the reverse. So this is what the influence of the class A gene having the apetala2 in the normal form is going to develop the normal organs. But as it is a recessive mutated gene, it is going to give the other oles a chance to develop. So here this is what it is going to appear like. Now here mutations in type B genes. In this, it affects the petals and stamens, which are the intermediate holes, that is the hole 2 and the hole 3. So here two gene mutations have been found. Apetala3 and PISTILLATA represses the development of the sepals instead of petals and carpels in the place of stamens. Now when you are going to observe here, this is Apetala3 and PISTILLATA. Apetala3, there is the absence of the petals. PISTILLATA, there is the absence of the stamens. So these two genes will be responsible for the development of that particular organs, the petals and the stamens. Now here when you are going to consider this organ, this is the sepal. In the place of the petal, the sepal has developed. Now this is the actual place of the carpel. In the place of the stamens, the carpel has developed. So you can observe the mutation. So here it is meant that the apetala3 and PISTILLATA controls the development of the petals and the stamens. Similarly, here apetala2 controls the development of the sepals and the petals, getting influenced with the apetala3. So this is what here and here. Now here the formula changes again in the apetala3 and the PISTILLATA. So here there will be the sepals, sepals, carpels, 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 sepals, and sepals. So this is what the thing. Now again, mutation in type C genes. In this, it affects the reproductive hole development, namely the stamen and the carpels. In this mutant, the gene is AGAMOUS, replaces the development petals instead of stamens and sepals instead of carpels. So here it is going to replaces the petals in place of stamens and sepals in place of the carpels. Now if you are going to see the picture, sepals, petals, petals, sepals, petals, again petals, sepals, petals, sepals, sepals here in the groups, sepals, petals, petals, sepals, and it is going to be the again the same. Now here in this all three mutants, we have observed that a specific gene is going to get mutated. Because of that, there will be the abnormality observed within the two different adjacent holes. So here how it can be understood. Here in the first mutant, that is APISTILLATA2, that specific gene will be APISTILLATA2 will be responsible for the development of the sepals, and it also be responsible for inducting the boundaries of the petals and also inducting the development of the petals. But in within this, what is happening, apetala 3 is normal in its function, you can see, but it is not expressing because due to the abnormality function of the apetala 2. So as we have discussed that due to the mutation within a gene, the two different adjacent holes are going to get affected. So here within this apetala 2, there is the effect of this apetala 2 where there is no development or the underdevelopment or suppression of the development of the sepals and the petals. So in this place there is the influence of the dominant normal genes of the corpus and the stamens. In the same manner, if you are going to consider the apetala 3 and pistillata, here also there is the absence of the petals and the stamens. So here it has got influenced by the development of the sepals and the corpus. In the same manner, here also due to the mutation in the AGAMOUS gene, there is the influence of the normal genes which are going to develop the sepals and petals in the place of the stamens and the corpus. So this is what the particular description. Now in apetala, it gives the meaning of the absence of the outer holes, that is apetala 2 will be responsible for the abnormality within the sepals and the petals. Apetala 3 and pistillata, here it is going to give the meaning of the abnormality in the petals and the stamens, and the gene mutant the AGAMOUS is going to cause the abnormality in the development of the stamens and the carpel. So this is what the four different homeotic genes will be responsible for the development of the flower. Okay guys, this is what the explanation of the development of Arabidopsis thaliana, where the genetic mutations describes the specific difference in the development of the flower, which is going to give the complete understanding that how the flower is developing with the help of the ABC model of the flower development, where the organ identity genes has the influence on the development of the different organs of the flower, and again whereas the organ identity genes has been influenced by the homeotic genes during the flower development. So this is what the complete explanation. I hope I had been very clear during the explanation and audible to you guys, and I think it is very clear for you to understand this concept through these slides. And thank you for tuning to the Learner's Big Book, and I request you to spread these slides and also help me with your required topics on which I can develop the slides, so you can make use of that particular knowledge. And I also request you to give me the feedback about any cons or the pros within the explanation, and also help me with your good opinion or any bad opinion also about these particular developed slides and also the way of explanation. And I thank you for your kind help for watching and spreading these particular slides. Thank you guys.